Imagine for a second that you have this perfect living digital twin of a major city running on your laptop.
Right, like a fully functional simulation.
Exactly, not just a static map of the roads, I mean a living simulation. You can see the exact movement of every single car, the pedestrians on the sidewalk, the traffic lights changing.
Oh, that's a cool way to picture it.
Yeah, and it's all rendered in real time, totally governed by the actual laws of physics. Now, I want you to take that entire concept, that level of insane dynamic detail, and scale it down to the microscopic level.
Down to a single cell.
A single human cell. Which brings us to the mission of today's deep dive. We are unpacking a genuinely groundbreaking 2026 paper from the journal Cell, published by Arkfeld and colleagues.
It really is a staggering piece of work when you look at the details.
It really is. We're going to explore how these scientists actually built a particle-based, four-dimensional digital twin of a living cell. And they did this to crack the code on how mitochondria, those little powerhouses of the cell we all learned about in high school.
Right, the classic powerhouse analogy.
Exactly. They wanted to see how they actually move, shape-shift, and react when put under stress. So, okay, let's unpack this. Because this isn't just a niche topic for cell biologists. This is about a revolutionary new way to simulate life itself.
It absolutely is. But to really appreciate the leap they've made here, you kind of have to understand the historical roadblock we've faced with what we call whole cell models, or WCMs.
Right, because people have been trying to simulate cells for a long time, haven't they?
For decades. But they were trapped between two extremes, basically. On one side, you had these static, frozen snapshots. So techniques like cryo-electron tomography, which give you this incredible atomic-level structural detail.
But it's frozen.
Exactly. The cell is frozen. It's a gorgeous photograph, but it's completely dead. You can't see how things function over time.
It's like taking a snapshot of a highway. It doesn't show you the traffic moving. It just shows cars parked on the road.
That's a perfect way to put it. Then on the other extreme, you had these massive numerical spreadsheets like flux balance analysis.
Okay, what does that mean exactly?
Well, it tracks chemical inputs and outputs perfectly, but it has zero physical shape. It's really just a mathematician's ledger.
Right.
So what was completely missing from science was the mesoscale.
The mesoscale, meaning like the physical reality of how all these squishy living organelles actually crowd each other.
Exactly. How they navigate around the nucleus, how they bump into membranes over a span of seconds or minutes.
But wait, hold on. If the problem is just that we need to see movement over time, why not just, I don't know, record a video of a live cell? Like put a cell under a powerful microscope and hit record.
It seems like the obvious solution, right? But it comes down to the physics of traditional microscopes. Think of traditional confocal microscopy, like shining an incredibly harsh, blinding spotlight directly into someone's eyes.
Oh, wow.
Yeah. To get the high-resolution, three-dimensional data you need for a model like this, you have to pump so much laser light energy into the cell that you literally burn it.
You burn it away.
Yeah, it's a phenomenon called phototoxicity. The cell gets stressed from the heat and the light, its behavior fundamentally changes, and it just starts to die before you can extract a meaningful 4D model.
So the very act of observing the cell kills it. You can't model healthy traffic if you're actively setting the city on fire with your camera.
Exactly. It's a hugely frustrating catch-22 in biology.
So how did this team get around the phototoxicity problem? Because clearly they didn't burn these cells.
No, we didn't. And that is where the technological breakthrough in this paper begins. They utilized a technique called lattice light sheet microscopy, or LLSM.
LLSM. Okay, how is that different from the harsh spotlight?
Instead of blasting the cell from the top down, LLSM illuminates the sample from the side with a microscopic, ultra-thin sheet of structured light. It essentially sweeps through the cell optically, slicing it with this incredibly gentle light.
Slicing it with light? That sounds like sci-fi.
It really does, and it captures a full 3D volume every single second, with minimal photo damage.
Wow, so it's capturing 4 dimensions, X, Y, Z, and time, without fying the subject.
Exactly. And the resolution numbers they cite in the paper are just wild. It's about 250 nanometers laterally and 500 nanometers axially.
So they are getting high-definition 3D video of the cellular guts in real time, and the cell doesn't even know it's being watched.
Right. And because the cell stays perfectly happy and healthy, you can watch the organelles go about their normal, unstressed business. This is what gave the researchers the massive mountain of raw, moving data they needed to finally bridge that mesoscale gap.
Okay, so gathering our thoughts here before we move on, just to recap for you listening. The big historical problem was that whole cell models either gave us dead snapshots or just pure math without a physical shape.
Right.
Traditional microscopes couldn't solve this because they literally burn the cell alive. But this new gentle light sheet technology lets us safely film a high-def 3D movie of the cell over time.
You nailed it.
Which leads to a massive question for you listening at home. Think about how much processing power your phone needs just to render a 3D video game. Now imagine rendering the chaotic, squishy guts of a living thing. How on earth do you translate a glowing, microscopic movie into a functional mathematical simulation?
Yeah, that translation is where the computer science truly meets the biology. To build this simulation, the researchers used Cal-27 cells.
Okay, well, what are Cal-27 cells?
They're an oral squamous carcinoma cell line. And what they did was stain the mitochondria with a fluorescent dye called mitotracker green. Then they stained the microtubules, which are basically the long structural highways of the cell, with tubule and tracker deep red.
Okay, so we have our visual canvas. We've got glowing green powerhouses moving along magenta highways.
Exactly. And what's fascinating here is their isolation strategy.
Oh.
Because they knew that if they tried to simulate the incredibly complex traffic system of the cell all at once, the math would just be impossible to untangle.
Too much going on at once.
Right. They needed a baseline.
Yeah.
They had to understand how mitochondria move when the highways are completely shut down.
Interesting.
So before they did anything else, they treated these Cal-27 cells with a drug called nocaduzole for 60 minutes.
Nocaduzole, what exactly is that doing to our glowing canvas? Is it like paralyzing the mitochondria?
Not the mitochondria directly, no. Nocaduzole depolymerizes the microtubules. It basically dissolves those magenta highways.
Oh, wow.
Yeah, so after 60 minutes of this drug, the tracks are entirely gone. And without those tracks to attach to, the mitochondria just kind of float around in the cellular fluid. They passively diffuse, bumping into things randomly.
So nocaduzole is essentially a chemical wrecking ball for the cell's road system. And by doing this, they've isolated just the passive floating movement.
Precisely.
But how do you take that video of floating green blobs and turn it into actual math?
Well, they fed that visual data into an advanced simulation software framework called READY. And this is really the technical marvel of the paper.
READY, spelled with a capital R, a small e, a small a, and 2 capital D's, right?
That's the one. READY takes the continuous visual glow of the mitochondria and breaks it down into a discrete particle-based graph. They modeled the mitochondria as tiny individual spheres or particles with a very specific radius of 0.15 micrometers.
Wait, why 0.15 micrometers? Is that just a convenient number for the computer to process?
Not at all. That measurement perfectly matches the real-world spatial distribution of mitochondrial DNA nucleoids. They quite literally mapped the actual physical geometry of biology into the code.
Okay, but a mitochondrion isn't just a single perfect sphere. Every textbook shows them as these long, stringy, almost bean-like networks. If the computer just sees them as individual 0.15 micrometer balls, how does it simulate a long, continuous string?
That's the clever part. To make these individual spheres act like a unified, flexible network, the READY software links them together using what are called harmonic bonds.
Harmonic bonds, like tiny springs.
Exactly. Think of them as microscopic mathematical bungee cords holding the spheres exactly 0.3 micrometers apart. If they get pulled too far, the simulated spring pulls them back.
That makes total sense.
And they also use the visual data to build rigid, fixed boundaries for the nucleus in the center and the outer plasma membrane. That way they ensure the simulated mitochondria couldn't just, float off the screen into empty digital space.
I see. So they have these chains of spheres held together by mathematical bungee cords trapped inside a digital balloon.
Yes, exactly.
But mitochondria aren't just static strings. They're constantly shape-shifting, right? They snap apart, which biology calls fission, and they merge together, which is fusion. How do you program a digital bungee cord to organically snap and reattach like living tissue?
Well, you have to apply strict, probability-based topological rules. Let's look at fission first when the string breaks. In the READY simulation, fission is treated as a random, spatially independent reaction.
Spatially independent, meaning it doesn't matter where it is in the cell.
Right. The software assigns a specific probability to every single bungee cord in the chain. Specifically, they programmed a bond removal rate of 0.000044 per second.
So it's like every fraction of a second, the software is rolling a massive multi-sided die for every single link in the chain. And if the die lands on the wrong number, that specific bond just vanishes and the mitochondrion splits in two.
You've got it. And that 0.000044 rate perfectly matched the actual fission rate they tracked in the living cells.
That's incredible.
Yeah. Now, fusion, when two mitochondria join together, that works differently. Fusion is spatially dependent. If two separate mitochondrial particles are floating around and they randomly drift within a specific threshold distance of each other, the simulation instantly snaps a new harmonic bond between them. They fuse.
So to recap this crucial step for you listening, to build a digital twin, the researchers first chemically demolished the cell's highways to isolate how mitochondria passively float and shapeshift. Then, they mapped that video into a particle simulation, creating chains of spheres connected by mathematical springs that constantly break and reform based on real-world probabilities, which gives us a perfect simulation of a sick, passive cell.
A completely passive baseline, yes.
But that begs the next question for you to think about. If you're the scientist looking at this passive digital twin, how do you add the complex highway system back in? And more importantly, how do you simulate the biological trucks that actually drive on those highways?
Yeah, that is the ultimate challenge for this model. The passive twin is just the foundation. Now, they needed to model the active transport.
Here's where it gets really interesting. They moved away from the no-codazole-treated cells and looked at untreated, perfectly healthy Cal-27 cells. The magenta microtubule tracks are completely intact.
But immediately, they hit a massive technical hurdle. A microtubule is incredibly thin, only about 25 nanometers wide, that it's so small that it is literally below the diffraction limit of the lattice light sheet microscope.
Meaning the microscope is incredible, but physics physically won't allow light to resolve a structure that tiny?
Right. You can see the magenta glow, but you cannot perfectly see the exact physical lines of every single microscopic track. So, to solve this in the digital twin, the team developed a procedural generation method called ready-made models, or RMM.
Hold on. Procedural generation? Like how a video game algorithm will just spawn a random forest as you walk through it.
Sort of, yeah.
If they can't physically see the tracks and they're just letting an algorithm generate them, aren't they just guessing where the highways are? How is that an accurate twin?
That is a very fair point of skepticism, but it's not just a random guess. The RMM algorithm used the spatial intensity gradients from the actual imaging data.
What does that mean in simple terms?
It looked at the 3D cloud of magenta light. And where the glow was brightest and most concentrated, the computer procedurally drew a track right through the mathematical center of that intensity. It's tracing the most likely physical path based on the density of the light. And crucially, they polarized these digital tracks.
Polarized? You mean they aren't just two-way streets?
Exactly. Microtubules are not static ropes. They are highly directional. They have a distinct forward and backward. In a cell, the minus end of the track points inward toward the center of the cell, the centrosome.
Okay.
And the plus end points outward toward the edges, the cell periphery. The procedural generation respected this biological rule, giving every single generated track a one-way flow.
Okay, so we have one-way highways traced from the light gradients, but the mitochondria can't move themselves. They're just the cargo. You need a locomotive. And in biology, those locomotives are specialized motor proteins.
Right, the cellular engines.
Yeah, you have one motor called kinesin, which carries cargo outward toward the edges of the cell. That's anterior grade transport. And you have another motor called dynein, which hauls cargo inward toward the center, which is retrograde transport.
Exactly.
Actually, here's a way I'm going to remember this, and maybe this will help you listening too. Kinesin kicks out to the edge, and dynein dives into the center.
Huh? That is a perfect mnemonic. Kicks out and dives in. I like that. Now, modeling the physics of kinesin and dynein inside a computer is honestly a masterclass in computational biology. The researchers couldn't just tell the digital mitochondria to simply slide along the track.
Right, that wouldn't be physically accurate.
Exactly. They modeled the motors themselves as what are called two-state Markov processes.
I've heard of Markov processes in statistics, but how does that apply to a tiny biological motor?
Well, a Markov process is a mathematical system that transitions from one state to another, where the probability of the next state depends only on the current state. It has no memory of what happened 10 seconds ago.
Okay.
These simulated motors, the two states are simply active or inactive.
So it's like a microscopic goldfish. It has no memory. Every single millisecond, the goldfish rolls a microscopic die. If it rolls a high number, it reaches out and binds to the track. If it rolls again, it takes a step forward. If it rolls a low number, it just lets go.
That's a brilliant way to visualize it. They dynamically attach, step, and detach, purely based on these memoryless stochastic probabilities. And the researchers pull these exact probability numbers from real-world biochemical data.
So it's anchored in reality.
Totally. But they went even further. They programmed in force-dependent detachment.
What does that mean in practice? Force-dependent detachment.
Let's say a dining motor, the one diving in, is hauling a massive mitochondrion. But the mitochondrion gets snagged on the nucleus or tangled in another structure.
Like getting caught on a branch?
Yeah. In the simulation, the tension on that mathematical bungee cord builds up. If the pulling force gets too high, the mathematical bond actually breaks under the strain and the motor snaps off the track. The simulation mimics the actual physical breaking point of the proteins.
That is an astonishing level of fidelity. They aren't just moving pixels across a screen, they are actually simulating the physical forces and failure points of motor chemistry.
It's incredibly detailed.
So, stepping back for a moment, we now have a model where healthy cells have polarized one-way microtubule tracks, and we have explicitly coded motor particles, kinesin kicking out, dynin, diving in, that attach, walk, and snap off based on complex math and physical tension.
Right.
Which brings us to the most critical question of this entire deep dive. We have a twin that mimics a healthy cell and a twin that mimics a cell with no tracks. How do we prove this computer program can actually predict biology rather than just blindly copying what it was fed?
That is the dividing line between a fun animation and a true scientific model. If this digital twin is scientifically valid, it shouldn't just replay the video you used to build it. It needs to predict how the system will behave under a completely new, unseen condition.
The ultimate predictive test. So the researchers took a totally new living Cal-27 cell, and they treated it with that highway-destroying drug, no-cortisol. But this time, they only treated it for 30 minutes.
Exactly. At 30 minutes, the no-cortisol hasn't finished the job. You have a partially destroyed highway system. The microtubule mass was cut roughly in half.
So how do they test the digital twin against this half-destroyed cell?
They fed the simulation the initial half-destroyed starting state of this new 30-minute cell. They gave it the broken tracks, but, and that is the critical part, they kept all the underlying math exactly the same as the healthy cell.
They didn't change the rules.
Right. They kept the motor speeds, the goldfish die rolling probabilities, the binding rates, all identical to the healthy control cell. They didn't tweak the math to make it fit the new scenario. They just said to the computer, here is a broken highway system. Use the rules of physics we gave you and show us what happens.
And what happened?
The result was a complete triumph. The digital twin perfectly predicted the intermediate motility of the mitochondria. Specifically, it matched a metric called the mean squared displacement.
Mean squared displacement, or MSD. For those of us who aren't biophysicists, imagine tracking a person who is wandering aimlessly in a giant field. The MSD is basically a measurement of how much total area they manage to cover over a given amount of time. It's a gold standard way to measure overall motility or movement.
Exactly. In the healthy 0-minute cell, the MSD was high, lots of rapid movement along the tracks. In the 60-minute no-cortisol cell, the MSD was low, just slow, passive floating.
And the 30-minute one?
The digital simulation of the 30-minute cell predicted an MSD right in the middle, and it perfectly matched the biological reality of the actual live 30-minute cell. The twin proved that the physics held up without needing to be manually tweaked or reparameterized.
That gives me chills. So to summarize this victory, the digital twin was tested on a partially disrupted cell. Without cheating or altering the underlying math, the simulation accurately predicted exactly how much area the mitochondria would cover on those broken tracks.
It's a true predictive engine.
It's a working sandbox of human biology, which leads to our next prank for you. You now have this perfect digital sandbox. What happens when you use it to simulate a cellular panic attack?
This is where the paper stops just validating its model and moves into actual scientific discovery. The researchers decided to use their sandbox to investigate a massive biological phenomenon called paranuclear clustering.
Paranuclear clustering. It's a really dramatic stress response. When our cells face intense, life-threatening stressors like hypoxia, which is a severe lack of oxygen, or a viral infection like hepatitis B, or massive genotoxic DNA damage.
The really bad stuff.
Yeah, the really bad stuff. The mitochondria do something wild. They stop whatever they're doing, abandon the edges of the cell, and rush to the absolute center, clustering tightly around the nucleus.
And if we connect this to the bigger picture, you have to understand the canonical theory behind this. For years, biology has operated on a motor-centric assumption. The belief was that under stress, the cell simply cranks up the dynin, the inward diving motors, and turns down the kinesin.
The motors just overpower everything and drag the mitochondria to the center.
Exactly. That was the assumption.
So the researchers put this age-old assumption to the test in the digital twin. They programmed in these stress parameters. They shifted the dynein to kinesin binding ratio drastically to 10-to-1. They bumped up dynein activation by tenfold. And sure enough, the digital mitochondria swarmed the nucleus.
The simulation showed their average distance to the nucleus dropped from 6.6 micrometers down to 4.19 micrometers.
Right.
It completely confirmed the canonical theory. The motors can do the job.
Well,
But here's the plot twist. Because they have a digital twin, the researchers could ask a deeper mechanical question that you could never ask in a living cell. Is it just the motors driving this rush to the center? Or does the physical, structural architecture of the track itself play a hidden role?
to test this, they ran a topological perturbation experiment, and this blew my mind. In the computer, they kept the exact same stress parameters, those super-powered 10-to-1 inward-driving dynein motors. They kept the exact same total mass of magenta tracks, but they virtually bisected every single microtubule.
They just digitally chopped the tracks in half.
Yeah, so the tracks went from an average of 4.34 micrometers long to 2.17 micrometer long. The cell still had the exact same amount of track material, it was just chopped up into disconnected, shorter segments.
And the result of that single structural change was astonishing. The paranuclear clustering completely stopped.
Stopped. The mitochondria got trapped on the periphery of the cell. Their distance from the nucleus stayed stuck at 5.34 micrometers. I want to make sure we truly grasp this. Even with those super-powered dynein motors working in absolute overdrive, screaming to get to the center, they couldn't reach the nucleus.
The physical topology of the microtubules acts as a structural gatekeeper. It completely overrides the motor chemistry.
It's like putting a 1,000 horsepower Ferrari engine inside a train, but you've ripped up the train tracks and left them in disconnected 10-foot segments. It absolutely does not matter how powerful your engine is. If the physical track doesn't connect to your destination, you aren't going anywhere.
That is the perfect analogy. And it proves a massive paradigm shift. The physical network connectivity of the cell matters just as much, if not more, than the motor chemistry powering it.
Let's gather this monumental finding together. Stress makes mitochondria rush the cell's center by ramping up those diving dynein motors. But cutting the digital tracks in half prevented this clustering entirely, proving that the physical length and connectivity of the tracks are hidden gatekeepers dictating cellular behavior.
It's a huge revelation.
Which leads to our final question for you listening. If you are a massive pharmaceutical company trying to design a drug to stop a disease, where should you actually be looking?
That is the exact billion-dollar implication of this paper.
Yeah.
We have spent decades in pharmacology focusing almost entirely on chemical kinetics. If a transport pathway in a cell is failing, our default response is to design a drug to stimulate or inhibit the motor proteins.
So what does this all mean for us? It means if you are trying to treat a disease linked to mitochondrial dysfunction, and we are talking about massive, devastating global health burdens here, like diabetes, neurodegenerative diseases like Alzheimer's and Parkinson's, and various severe cancer.
Yeah, major killers.
Right. You might be targeting the entirely wrong mechanism. If your new wonder drug targets the motor proteins to fix a transport issue, it will completely fail if the underlying highway topology of the patient's cells is fragmented.
Precisely. And this whole cell digital twin framework finally allows scientists to run these complex mechanical experiments in a computer before they ever synthesize or test a drug in a human trial.
That saves so much time and money.
Oh, absolutely. They can map the geometry of a diseased cell, perturb the digital topology, and actually see if the physical, structural architecture of the cell will even permit the drug to work.
It's like having a crystal ball for physical biology.
This raises an important question, really a philosophical one for all of biology. We're taught to think of cellular biology as this floating soup of chemical reactions and concentration gradients. But this deep dive proves that the physical architecture, the microscopic shifting geometry and physical tension of the cell, is a hidden control variable.
It dictates life, death, and disease just as much as the chemistry does.
Without a doubt.
Which leaves us with a final thought for you to mull over as you go about the rest of your day. If the microscopic physical architecture of a single cell, like the sheer length of its internal highways, dictates its chemical fate, what other hidden gatekeepers exist in our bodies that we haven't even thought to map yet? What other physical structures are overriding our biology, just waiting in the mesoscale for a digital twin to finally shine a gentle light on them? Thank you so much for joining us on this exploration of the digital cell. We'll catch you on the next deep dive.